Okay, so let me show you something. Okay, I'm just closing this one up to here. So when you click show, go to the show and just click the cartoon. Okay, if you click the cartoon, you can find the protein in you can able to rotate the protein in 3-dimensional Armstrong region, however you want to see. This is a protein of a before you have seen that the protein was in the primary sequence level. Now the protein is in second tertiary structure level. Okay, it has alpha sheets, it has beta sheets. Those all things are combined to form a tertiary structure. So I just want to remove these lines. Some lines are available here. I just want to remove them. So what I shall can able to do, go for hide selection, go for hide, click hide, select lines. Okay, just click. It has disappeared. Okay, you can also hide the representation of this particular cells also if you want to remove them, because those are water molecules, that's why those are not removed. So this is the representation. I want to represent the color representation of each and each and single chain in different colors. So what shall I do? I'll go for the C for color. Go for color, go for chain. So if you select the chain, you will able to represent each and every single chain with different color. If not, I just want to select all things in a single color. You can go for color and go for by SS. Those are sheets. Okay, helix and beta sheets. Just click this one. You will represent the color representation will be changed. Okay, this yellow color representation called beta sheets, and the red color one is called alpha sheets. Okay, are you able to following me?
Yeah.
Okay, now I just want to remove the A chain and B chain or something called C chain. Any two of the chains I just want to remove it. Okay, if I want to remove any two chains, just go for this particular type of heading. Okay, just go for that particular heading, go for display, select the sequence. Are you able to see this one? There is a sequence has came here, appeared. I will change the representation also. Now you see the total green was some A chain. Okay. Total gray color was a B chain. Okay.
Yeah.
And the other pink one was a C chain. Can you see? There's a difference. Okay. And I want to remove B chain. Okay. I want to remove B chain. So just do the simple thing. Before we were having some difficulty in removing the chain. Okay, just selecting the everything and so going for the starting chain and going to the ending termination. This made us to feel difficult. So I'm just selecting here. Just give the left click and select it everything. If you select the entire protein will be selected. Are you getting what I'm saying?
Just
Are
to
you able to
select
follow me?
the sequence to delete that particular chain, am I right?
Yeah, right. See, I'm selecting the entire protein. It is selected. So this is. Are you feeling any difficult to select in this way? This is the way.
No,
Is
sir.
there any
No.
no difficulty? So this is the very easiest way to eliminate the protein. So see, everything will be selected. So everything will be selected till the ending. You can able to select. So let me select the everything.
Sir, while
After
you
that,
selecting, it is going to deleting, no?
because we want only single chain. There are 3 chains are available to this particular protein. You want to prepare your protein for the before you go for docking. So you are not going to dock to all 3 proteins. You are going to dock only at particular place. That's why we are selecting. Before without preparing the protein, how you going
A
to do dock?
particular active site like that, you mean?
Not the active site we are deleting. We are not. It is not required, or because these are twin proteins. We don't want twin proteins. These are not twin protein. 3 proteins are similar, but when 3 proteins are similar, I want only a particular region of the protein. Remaining things I can able to remove it. I don't want.
Particular region or particular protein?
Particular protein which is not useful for me, I'm removing them.
Okay.
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 So one second.
But in that case, we could have downloaded the particular needed protein from the PDB site itself, no?
But your protein doesn't have needed protein, then what you do? That is only having the 3 proteins. Then what you do? 3 chains are there.
3 proteins or 3 isoforms?
Not these are isoforms. These are 3 chains.
3 chains of the same protein, you mean?
Yeah, same protein of 3 chains.
Okay, that's what I asked. The particular chain is
Oh,
what
for
is the area of
example,
interest?
you have 3 chairs of similar one.
Okay.
You have an option to sit. You are the only person want to sit in a chair.
Okay.
If 3 chairs are similar, which chair you are going to choose?
Depends upon the best fit.
Best fit. Or else, if it is all are fitted the similar one, if all are fitting the same method, then you are going to choose only one. Remaining two you are going to keep other place, right?
Yeah.
Here also we are doing the same thing.
Okay, got it.
We are not taking all 3 proteins. If 2 chains are having something different, then we will generate 2 grids, 2 active site place residues, 2 binding site residues will be generated at a similar place only. Okay.
Okay,
But
fine.
here they are similar. That's why we are removing. Okay.
Okay, got it.
And I'm also removing this. These are there is something called ion. There is 0 here. Can you see the 0 here?
Yes.
That 0 is also called as water molecules. Okay. Water, sometimes we call as tails. So this doesn't plays a action role in our docking. That's why we don't want these all things. So I'm selecting entire thing, just selecting the entire thing. So just give right click, remove. Can you see now? Is it very easy to do?
Yeah.
Are you
Yes.
feeling? Is there any difficulty to do?
No.
No.
Okay. There is some dots are available. Okay. Some dots are available. I also remove them. You need to observe something different when I'm doing everything. So at later on, you can able to understand what I'm doing. So I have removed everything now. Got it? Hmm?
Yes.
I have removed everything. I want to see something called the particular region. I want to see the ligand residues. What is the ligand residue for particular region of Q39? I selected Q39. See here, there is a selection. The selection shows I selected here Q39. Okay. I want to see here the 4 angstrom regions of that particular protein. Okay. If I want to see the 4 angstrom region of the particular active site of the Q39, okay, I
The
just go
Armstrong
for,
region is chosen based on resolution.
yeah, no, resolution, it's not resolution. For every protein, it has some particular angstroms. Those
Okay.
particular 4 angstrom region doesn't have some conformation, pocket chain. We are finding the pocket chain, default pocket chain. If I want to dock at this particular ligand as a control, I want to see how far my proposing compound or proposing drug compound is going to binding with the efficacy. How far it is having efficacy. Just want to see with the which is already there in my protein data and my target. I'm just seeing there. So after you are selecting the Q39, okay, just go for action. Okay, select the modify around residues within 4 angstrom region. See, after I'm selecting the 4 angstrom region, the entire selection has been selected. Did you observe anything difference?
Yes, sir.
After that, see, I want to label those residues. What are those residues? Okay, so let's go for label. Go for select for residue. Can you see now residues are been selected?
Yeah.
Some so you may be seeing some publication. They will be showing some residues will be they show us at this region we have binded our some compound, antiviral compound or something. They will be noted. Okay.
Okay.
Okay, this is the representation of 4 Armstrong region of that particular protein. Okay, and I want to see only valine residues. Okay, I want to see only valine residues, or else I want to see a particular region of the protein. What I shall do? I just go here and write down the some code SELE select residue. I want to select. I want to see only particular region of those residues. If you want to see any particular residues, like if you hello.
Sir, where you are going to open that guided system?
Your voice is not audible, not clear. Can I just getting some reason? Can you talk to me once again?
Sir, is it audible, sir?
Yeah.
That dialog box, how did you open it, sir?
It will be automatically opened to you. When you open the Pymol itself, it will be there. See, this both will be there here only when you open. Just I have been just minimized. Just minimized. You see here it is already there. A dialog box. You will find the dialog box always.
Can I check? Just a minute, sir. Just a minute.
You just install and open it the PDB ID which you want to open. Then it will be automatically opens. No need to open once again for this particular dialog box. There is no other option to open second time. Okay.
Okay.
So what I'm doing, select residue. I want to select something called let me see what residues are common here. Shall we select lysine? Okay. There are L is common in everything, right? So I'm just trying to select for that particular place. I want to show that one. So I selected lysine. So I think I selected leucine, not lysine. It has went something. So now total L's are selected, right? Can you see? All L's are selected. Preeti ma'am, Kasturi ma'am, Arvind, do you see any of the difference? I have selected L only. I have given some code to this particular protein. That is a default one. I
Yes,
have
sir.
selected the residue called L.
Sir,
It
now
has been
what you have
selected.
done in the Wattpad you have done, no?
Not in water. It have done in the dialog box. I have given in the dialog box.
Sorry, it's
This
not
came
clear.
from
Can you
Huh?
just maximize
where
the?
you have opened this?
Ma'am, when you open the file itself, software itself, you can open this particular dialog box.
Okay.
Sir, can you
I'll
just
show you later. I'll
maximize
show you
that
later.
one?
Okay, sir.
Okay.
Okay.
Sir, can you just maximize it?
Which one?
This screen.
Yes, I maximize it.
Not clear for me, sir.
I have done now. Can you see? Are you in desktop in mobile?
I'm using desktop, sir.
I have opened everything. What about you, Preeti ma'am and Kasturi? Are you able to see?
Yes, sir.
Yeah.
I don't know. You have to see once again, Arvind. I have opened everything. It is in maximize only. I have not minimized my
Okay,
desktop.
okay.
It's everything is a full screen only. Just clear it from your side once again. So I have given some dialogue. Okay, that is called select residue. Okay, select a residue. If I want to see valine, go for select valine. That is the 3-digit code on whatever you want. If you want to see glycine, give glycine. It will be selected everything. No need to go and find everything. Okay. After I'm selecting, oh, I'm sorry, it has been closed. Once again, I'll open. Where is this? Mm. Okay, let me do here itself without editing. So I just want to see something. I want to select lysine. Okay, I've selected lysine, only lysine. So you can also see only lysine will be selected everywhere. Okay, and also I want to see the spheres in. I want to select that particular lysine as spheres. You can see this, it has been selected the spheres. Okay, if you want to select those particular residues, go for show, select the spheres. Okay, or else if you want to select it as a mesh, you can select it in the representation, molecular surface representation, in the mesh representation. Okay, and if you want to, if you don't want that particular thing, after you just can able to hide it, it will get back. Okay, are you following me what I'm saying?
Yes, sir.
Yeah.
Okay, fine. I don't want to go for sphere also. I just gone back. If you want to delete any particular region, just go for here, select this particular region, just select the right click and go for remove. You can able to remove that particular region. Okay. Or else you want to see any particular region of this residue. Okay. You have selected this residue. See, it is showing something. See, it is showing this representation. Or else I want to select alanine here. I want to see phenylalanine also residue. I want to see this particular residue. I want to see this residue. Want to see these all residues of 4 angstrom region. Just go here and modify around 4 angstrom region. You will find, and you can able to modify the residues number also. You can able to color them based on which you are interested. So you can see them, the color representation, because I have selected only in single chain. That's why it is showing like this. These residues, 4 angstrom regions residue or pocket chain residues tells us the grid generation. We can able to generate grid there when we are preferring to go for local docking. Okay. When you are going for global docking, it is not required to go for pocket chain. You can able to dock by selecting the entire protein. But in case when you are going for particular region, okay, if you want to bind only at that particular site of that particular region of that particular amino acid, then you can able to find the pocket chain. Are you able to clear what I'm saying exactly? Are you getting what
Means
I'm
the
saying?
packet chain is required only for global.
Not for global, it is for local docking.
Okay.
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 It is used in autodock and pyrex also. In case of global docking, the entire protein will be selected. Then we are not going to look for the residues.
Okay. Okay. Okay.
When a particular region of that residues only we are concentrating, then we call them as local docking.
Means like that score matching will be done.
Score matching will be done the number of repetitions we do. It will be shown you in the next week session, ma'am.
Okay. Okay.
When we go for demonstration on the autodock.
Okay.
Okay, Arvind, is it clear for you also? And what about Kasturi, ma'am?
Yes.
Are you clear?
Yes.
So is there any doubt in doing this particular Pymol hands-on demonstration while editing the chains, or when you're selecting the Armstrong region, or when you're selecting the particular pocket chain identification, or you want to see entire how many lysine residues are there, and when you're selecting the color representation, these all things, there is any difference? There are doubt?
No.
What about you, ma'am, Preeti, ma'am?
No, actually, only
Are
the
you
problem is in installing the thing. But I got
it's
the
not
clarity.
no issue, ma'am. Once again, you do it. If it is
Okay.
not coming, we will connect once with a team viewer, then we'll sort out. Okay.
Okay. No problem.
And after that, see if you want to change the background.
Hmm.
Someone wants to go for publication. Okay, they want to go for publication. They are not interested in. See, we have one thesis. One of my senior thesis. I'll show you that one. PhD. Hmm. Okay, not an issue. I'll show you in some other thing. This only. PhD support letter. So it's not available here also. Okay, no problem. I'll show you later. So if you want to represent it this picture, okay, you can also give a code called BG. Okay, can you see here down at the arrow at the cursor I'm showing to you? Select BG background white. It will change BG. Whereas you want to go for yellow, select yellow. Whereas if you want in gray, you can select. You can able to take screenshot of this particular protein. Go for screenshot and go for that's it. You can go for publication purpose if you want to take some pictures and represent it based on the how you want these things. Is it clear?
Yeah.
Okay, then tomorrow when you're coming to the class, just go for downloading Cluster. Okay, Cluster X. Okay, just go for Cluster X. Type Cluster X in the Google. Go for download Cluster X. Okay, as have you done installation for Pymol? You can able to download it. Available for download here. Just click this one. Just download the Cluster X 2.1 MSI. This one. Okay, just click this one. Will be downloaded the Cluster X. Okay.
Yes.
Yeah.
Yeah, fine. Is there any doubts you want to ask? Tomorrow we will have homogeneous similarity searching using blast tree and sequence alignment using ClusterX software.
Sir,
Yeah,
sir, using MegaX software, can we, like, instead of downloading Cluster W or Cluster X, can we use MegaX software directly? We can see, is there a possibility like that, or we have to download it separately?
yeah, you can go for Mega also, not a issue. But for only for sequence alignment purpose, I'm using ClusterX. If in case if I want to do more than sequence alignment, like building a tree or building more than 3-4 sequence to want to see any phylogenetic analysis, then I choose to go for Mega. What you have said.
Because I have MegaX, so tomorrow directly can I use from that? I'm asking.
You can also use, but you can't generate in Mega the file formats. Some file formats like a PIR file format, protein information resource file format, these things. That's the problem. So I will show you something. What's the difference between cluster and this one? So can you see this one, the cluster X? This is the cluster X. So when you go for output format option, can you see these are the output format option? In Mega, only Mega will be available. But here, NBRF, National Brain Research Funding Resource like PIR format, DCG format, cluster format, FASTA format, these never generates. For when you want to generate a three-dimensional structure, it is required as that PIR format is required. This PIR file format file is required. Got it?
Okay, okay.
That's why I'm asking you to go for cluster. Okay.
Okay.
Okay, if you have any doubts, you can ask me, or else rest of the people can leave.
Is it the continuation of the Pymol?
Yeah, after the Pymol, this is a demonstration for Pymol, and we are going to have tomorrow the sequence alignment and sequence similarity search. How to search a protein similarity search even if it doesn't have a receptor, a target sequence. We are going to find a template tomorrow.
Okay, will the further classes be dealing with repository preparation and pharmacophore things?
No, pharmacophore we don't have in our syllabus.
Okay, no problem.
We are going to have only three-dimensional structure designing and protein validation. Later, we go for docking with the Autodoc Vina and Pyrex virtual streaming software. Then we go for 2D interaction images. These
Yeah,
all things
okay,
we will do.
fine, fine.
But pharmacophore is not we are going to do here. Okay.
Okay, okay.
Mm. Okay. Then I'm